Hello, hi, I welcome you to another educational video, and today's topic is C4 cycle or Hatch-Slack cycle, and this is a part of photosynthesis chapter in standard 11 NCERT biology book. And the C4 cycle, why the name is C4? Here the first stable product is oxaloacetic acid, which is a 4 carbon compound. That's why the name is C4, and it is discovered by Hatch and Slack. That's why Hatch-Slack cycle it is said. Now here what happens, first this atmospheric CO2 is accepted by PEP, that is phosphoenol pyruvate, that is the first carbon dioxide acceptor. And here this PEP, phosphoenol pyruvate, is a 3 carbon compound, and here 1 carbon is received, so it is forming the first stable product, that is OAA, that is oxaloacetic acid is the first stable product. Now here you see why it is written HCO3 minus. Now you see this atmospheric CO2 plus H2O, it's forming H2CO3, and when it undergoes breaking, so it forms H plus plus HCO3 minus, and this is written here HCO3 minus. Now after the formation of first stable product, that is oxaloacetic acid, this gets converted into a 4 carbon compound, that is malic acid or aspartic acid. Now this malic or aspartic acid gets transported to bundle sheath cell. Here you see in the mesophyll cell phosphoenol pyruvate is there, and here this is the first carbon dioxide acceptor, and in the presence of PEP case, that is phosphoenol pyruvate carboxylase, it is forming oxaloacetic acid. Here Rubisco is absent in the mesophyll cells, but in the bundle sheath cell there is Rubisco, you remember. And here the malic and aspartic acid in the bundle sheath cell, it is undergoing decarboxylation process, and here CO2 is released, and this CO2 undergoes C3 cycle. That means in C4 cycle 2 cells are involved. In mesophyll cell it is occurring the first step, and in the second step C3 cycle will occur in bundle sheath cell. Now after the decarboxylation from malic or aspartic acid, there is the formation of C3 acid, and this is pyruvic acid, and the C3 acid gets transported to mesophyll cell, and it undergoes regeneration where 2 ATPs are used, and in this process again phosphoenol pyruvate is regenerated to continue the cycle. Once again let me summarize the things. Here in the mesophyll cell phosphoenol pyruvate is the first carbon dioxide acceptor, and it forms the first stable product, that is oxaloacetic acid. Then it gets converted into malic or aspartic acid, which is a 4 carbon compound, gets transported to bundle sheath cell. In bundle sheath cell this malic or aspartic acid undergoes decarboxylation, and the CO2 which will be released, that will continue C3 cycle in bundle sheath cell, and after decarboxylation there is formation of pyruvic acid, which is a C3 acid, and this C3 acid gets transported and undergoes regeneration to form PEP again to continue the cycle. Now let us calculate the energy used in this process C4 cycle. Now here in the C4 cycle, for 1 CO2 fixation, two ATPs in the regeneration state, then 3 ATPs. You know this calculation from C3 cycle. So C3 cycle it is used. So total 5 ATPs. Now for 6 CO2 fixation, 5 ATPs into 6, that is 30 ATPs are used. Now come to 1 NADPH, 1 CO2 fixation, 2 NADPH, that is happening in the C3 cycle in bundle sheath cell. And for 6 CO2 fixation, there will be 12 NADPH used. Now here you see 30 ATPs and 12 NADPH are used. But in case of C3 cycle, so there was 18 ATPs and 12 NADPH are used. Now you come to the highlighted part of this C4 cycle. Here 2 cells are used. What are the highlights? Involvement of 2 cells. One is mesophyll cell, another is bundle sheath cell, and here the first CO2 acceptor is PEP, that is phosphoenol pyruvate. Then first stable product is OAA, so that is oxaloacetic acid. And some of the examples of C4 plant where this C4 cycle occurs, that is maize, sorghum, sugarcane, amaranthus, etc. Thank you very much.